Yeah,
Yeah. Okay. Okay.
yeah.
So, welcome to the afternoon session of this program. I'm Kavita Jain, and I'll be chairing this session. So, our speaker for today's session is Professor Joachim Krug from the University of Cologne in Germany. So, Joachim's research interests lie in both statistical physics and evolutionary biology. And before we start, I'll just remind you to off your cameras and mics. And if you have any questions, you can add that into the chat. And Joachim will give a couple of breaks in between, and you can ask these questions at that time. You can raise your hand or write in the chat. So over to you, Joachim.
Okay, yes, thank you very much, Kavita, for the kind introduction, and thank you, Debashish, and the other organizers for organizing this. So, of course, we all would have much preferred to meet in Bangalore in person, but I think this format also is nice. So what I would like to do today is to tell you a little bit about recent work that we have been doing on the problem of the evolution of antibiotic resistance. Now, of course, at the moment, we're all sort of very much concerned with another global health crisis related to the coronavirus, but the evolution of antibiotic resistance is also a major threat and a big concern for organizations like the World Health Organization. And the reason why this is such a big problem is that the evolution of resistance is a universal response of microbial pathogens, be they bacteria, viruses, or anything else, to essentially any biomedical intervention that you can think of. So also with the coronavirus, this will eventually become a concern. You maybe have heard about mutations that have occurred in these mink farms in Denmark, which might interfere with the vaccines that people are developing. So whatever you do to suppress a pathogen population, you need to be prepared that it will evolve resistance. And because this is an evolutionary process, because this is essentially Darwinian evolution, these evolutionary considerations are key for developing strategies that can prevent or delay resistance evolution. And that's of course why this is an important topic for research. At the same time, and this is more the perspective that I will be taking here, the evolution of microbial resistance is a model system that can be used to address broader questions of evolutionary theory, because one has relatively simple organisms subject to a very clear selection pressure that they have to deal with. So what I want to do in this talk is to discuss with you two case studies that we have done. The first one is an ongoing project, actually. So this is a system that we have worked on now for almost 10 years. This is a particular antibiotic resistance enzyme called TEM-1 B-lactamase that I will introduce in detail. And this is joint work with the experimental lab of Arjan de Visser at the University of Wageningen. And the second part will be about a different antibiotic called ciprofloxacin. And this is again a collaboration with an experimental group, in this case at the University of Edinburgh. So let me first introduce a little bit of terminology. So how do you quantify antibiotic resistance? Well, essentially, you apply the antibiotic to your bacterial or viral population, and you see how much the growth is being suppressed, right? So to quantify antibiotic effects, the standard method is to look at the so-called dose-response curve, which is the growth rate of the population as a function of drug concentration. And at some point, this growth rate will drop to zero, or go through zero rather. And so for larger concentrations, the population will effectively be killed. And this defines a so-called minimal inhibitory concentration, the MIC. So this is a quantity that will appear throughout the talk. And what a resistance mutation does is that it increases the MIC, right? So it makes the bacteria more tolerant to antibiotics. And the evolutionary pathways that I want to talk about here are pathways made up of multiple mutations, because often multiple mutations are required for the pathogen to reach high levels of resistance. And these multiple mutations then define an evolutionary pathway. So let me introduce the first main player of this story. So this is TEM-1 beta-lactamase. This is a protein which looks like this. And beta-lactamase means, so this is an enzyme that cleaves a certain substance, namely beta-lactam antibiotics. So this is an important class of antibiotics. The most prominent representative is penicillin, that you all know. And what these antibiotics do is that they target a certain molecule that the cell needs in order to build its cell wall. So they target cell wall synthesis. And so therefore, when this antibiotic is applied, the cell cannot form its wall, and it lyses and it dies. And TEM-1 beta-lactamase is a particular enzyme that confers resistance, particularly to ampicillin, to E. coli. This strange name. So it turns out this TEM is an acronym that derives from the name of the first patient, a Greek woman, in which this enzyme was identified in the 1960s. So this has been around for a very long time. And TEM-1 simply means, so this is a particular version of the enzyme. And so the typical experiments that are being done on the system consist in exposing this bacteria having this enzyme to a novel antibiotic, right? So the reaction to the evolution of antibiotic resistance, of course, is always that you try to develop novel antibiotics. So you try to modify ampicillin, for example, in some way, and that one of these antibiotics is cefotaxime. And so that initially the enzyme doesn't work very well anymore, but what then happens is, of course, that the bacteria adapt, and so the enzyme changes in such a way that it can again be effective against cefotaxime. So that's sort of the setting, and this is now a model system that is being used by several groups worldwide. And the starting point for my interest in this topic was a seminal paper by Daniel Weinreich and collaborators that appeared in 2006. And so they were the first to study systematically evolutionary pathways that lead to resistance of TEM-1 against cefotaxime. Now, this is a complicated picture, but it's very important, so I will try to spend some time going through it in detail. So each of these circles here is a particular version of the enzyme. So this one on the left here is the wild type. So this is the original TEM-1. And here on the right, you have a 5-fold mutant. So if you look closely, you can see that this has 5 minuses and this has 5 pluses. So in 5 positions in the gene, there has been a change in the corresponding nucleotide or amino acid. And the numbers below here are the MIC, that is the minimal inhibitory concentration in some units. And what you can see is that this 5-fold mutant is about 40,000 times more resistant than the initial type, right? So this is sort of, and the starting point of this experiment was actually this 5-fold mutant. So this was known from previous studies. It has actually never evolved in a clinical setting, as far as I know, but it was known to exist from a random genetic shuffling experiment. And so what Daniel Weinreich and collaborators were interested in was the question, how could this mutant, this 5-fold mutant, this highly resistant mutant, in principle evolve? And so to answer that question, they constructed all the intermediates between the wild type and the 5-fold mutant. So at every position, every one of these 5 positions, you can either have the wild type version or you can have the mutated version. And so that creates for you 32 combinatorial mutants, which all of them were created. This picture only shows some of them. So here you have a couple of first mutants. So this one has a plus in the middle. This one has a plus on the right. Here you have double mutants, triple mutants, 4-fold mutants, and here is a 5-fold mutant. And you can see the identity of the mutations written on these arrows. So for example, a very famous mutation is G238S, which replaces the amino acid G at position 238 in the protein by an S, right? And then there are some, so these are mutations that are denoted on the nucleotide level. This is because they are synonymous, so they don't actually, no, actually, this is not right, this is not synonymous, but this is a mutation that is actually not in the coding region. This is why it's a regulatory mutation. There are also synonymous mutations, but not in this particular example. And so you create all these intermediates, you measure their resistance, and then the question that Weinreich and collaborators asked was, well, if this fivefold mutant is supposed to evolve, it's probably most likely for it to evolve if each step on the way to the fivefold mutant is by itself beneficial, that is, increases resistance. And this is what you see here. So whenever there's an arrow, you see that it goes from a lower resistance to a higher resistance type, right? So this is a subset of the possible pathways that lead from the from the wild type to the fivefold mutant. So how many pathways are there? Where there are 5 mutations, and each pathway corresponds to a particular order in which these mutations occur. And so therefore, there are actually 5 factorial or 120 directed mutational pathways. So there are 120 different orderings in which these mutations could occur. And the key finding of this paper was that only a small set, namely 18 out of these 120, are monotonically increasing in resistance. And in fact, you can also assign a kind of weight to the different pathways, looking at the resistance differences that they cover. And this weight is encoded here in the thickness of these arrows, which are calculated according to different models. That's why they are sort of different colors. But the upshot is that there's only a small number of pathways that have appreciable weight. And this was sort of the main finding of this paper, which concluded that although there are many pathways, many possible pathways by which you could evolve from this wild type to the mutant, only a smaller subset of them is actually relevant. And this means that the process is more predictable, at least with hindsight, than you might have originally thought. Now, I emphasize here that these are directed mutational pathway where you, in each step, you add one of the mutations. In principle, of course, you could also have reversions. That is, you could add mutations at some of the sites, and then you could revert some other mutations. And because the effects of these mutations are strongly non-additive, this could lead to additional pathways. So this is a study that the same group conducted a little bit later, looking also at undirected pathways. So this is a similar picture of the same thing, except that now here you see some arrows going backwards. So this is now a situation where you have a double mutant, you add one mutation, you remove another one. And what you can see is that these sort of backwards path can help you to some extent to find a pathway towards higher resistance. And the total number of pathways is now an enormously large number. So this is essentially, mathematically speaking, the total number of all self-avoiding walks on the 5-dimensional hypercube, but only 27 of those are again monotonically increasing. Right. So this is sort of the setting. This is the problem that we want to understand. And I think it's clear that it looks like a rather appealing problem, not the least from the point of view of statistical physics. And so let me try to formalize a little bit this problem. But maybe before I do that, let me take a short break and just ask if there are any questions so far, if the basic problem and the framework is reasonably clear.
What do the colors represent, the color of the arrows when you sort the pathways?
In this picture, yes. So it turns out that what they measure are these MIC values, and that by itself is not sufficient to really, to really predict the probability of that particular step to be taken, right? So what they do is that they sort of create a kind of set of surrogate fitness values by simulations, which are ordered according to the ordering of the resistance values. And this can be done in different ways, and this corresponds to the different colors. So these are just different models, but the main pathways according to the different models appear to agree.
Thank you.
Can I ask a question?
Yes.
About the monotonicity. I have guessed that it's very important to be monotonous at the for the first mutations to escape from these states with low MIC. But once you have reached a certain threshold, you still need to have a monotonicity. You could, under the condition that you don't go back too much, you could still wander a little more.
Yes, I mean this monotonicity. It's a very strong restriction. It's for various reasons. It's of course not the whole story. I mean, one thing, as you say, it sort of depends on, of course, also what the concentration is that you need to adapt to, right? Whether you have a chance of growing, if you have a chance of growing, but maybe not optimally, then this might be tolerable. The other thing is, of course, that if the population is large enough, it can actually generate multiple mutations, and it could sort of cross these fitness values and so on. So there are sort of many reasons for why this is not the whole story. Why the reason why I nevertheless like it and will sort of stick to it for a little bit is that it's a very clean definition, right? So you can identify pathways of being monotonic or not without having to specify anything more about the actual dynamics. So it's sort of formally appealing. It's of course not the whole story. And I will get back to the issue of concentration dependence in the last part of the talk.
Okay, thank you.
So Joachim, a couple of questions from the students, or I mean other participants. One is TEM-1 is the drug. If so, then what does its resistance mean?
Let me see. Maybe I can actually. Can I see the chat? Yes, I can see the chat. Okay. No, TEM is not the drug. So TEM is the enzyme, right? So TEM-1 is the enzyme. So the resistance is the resistance. So what you do is, I mean, this is an enzyme that the bacteria can produce, and so you put the gene for this enzyme into the bacterium, and then you expose it to the antibiotic, and you ask at what concentration can the bacteria still grow. So this is how you sort of measure resistance. And okay, and then there's a question: what is the difference between directed and undirected pathways? Okay, so this is simply, I mean, I think this may become clear in the clearer in the next picture, but essentially directed means that you in each step you add, right? So here you have all minuses, here you have all pluses, and in each step you increase the number of pluses. This is directed. Whereas here you decrease the number of pluses and then you increase it again. So that's an undirected pathway, right? So there's a kind of directionality from the all minuses to the all pluses, and if you follow that directionality, then it's a directed pathway. Okay, is that clear? Okay, very good. So thank you for these questions. If there's no more questions, then let me go on. And so yes, so I want to put this into a mathematical framework. So this is sort of the picture, and here I'm sort of introducing now also the notion of a fitness landscape. So what I've shown you so far are, if you will, resistance landscapes. But of course, for a bacterial population exposed to these antibiotics, resistance is obviously an important part of fitness. So we'll, as I said in the last part of the talk, we'll be a bit more specific about the relation between resistance and fitness. But generally, the idea is, first of all, in a system like the one that I just described, you can encode the genotype, that is a genetic setup of your organism by a binary sequence. So you have a sequence of length L, where at each position you just denote whether the mutation of interest, like these resistance mutations, is present or absent, right? So this is a 0-1 variable. And if you put, and this is sort of now the graph on which this whole process appears. So if you put now, for example, for L equal to 3, if you put all the possible combinations of zeros and ones together, and you link them if they are separated by only one change, then you get this cube, right? So this is a 3-dimensional cube. And in general, you would have an L-dimensional hypercube. And then you can define a landscape on this space by just assigning numbers to these nodes, right? And so here, now there's a difficulty in sort of plotting a function on top of the space. So therefore, I've here indicated, let's say, the size of the resistance or the fitness by these circles. So this would now be the fittest version, and this would be the least fit one. And then you can orient this graph by drawing arrows in the direction of increasing fitness, and this then leads to the notion of a fitness graph, right? So a fitness graph is a hypercube where you put numbers on the nodes, and then you orient the links in such a way that the arrows always point in the direction of increasing values. And so now you can start to think about pathways. So in this particular case, you want to go from the all zero state to the all one state. And as I said before, each ordering of the numbers between 1 and 3 corresponds to one particular pathway. So this is, this would be one, and this would be another one, and so on. And in this particular example, you can see that all of these pathways allow you to follow the direction of the arrows, which means that in each step, you always go up, right? So the fitness always increases in each step. So this is a situation where all the pathways, all the 6 pathways are as we say, accessible, right? So this notion of accessibility is now the notion that pathways are increasing in fitness. And this is the case when, as we say, all mutations are unconditionally beneficial. This means that whatever, so if you do a mutation at a certain site, it will always increase your fitness, because what you can see is, for example, if you look at let's say the mutation at the first site, if you do that starting from a type which has all zeros, then you end up here. If you do the same mutation starting from a site that has a 1 in the middle, you go up here. So all these edges of the cube, the parallel edges of the cube, correspond to the same mutation occurring from different starting points. And if all these arrows point in the same direction, then all the paths are accessible. Now we can make it a little bit more interesting and just exchange 2 of these values, which has happened here. So now we have a bigger fitness here than here. And this then induces something that is called sign epistasis. So epistasis is a term in population genetics, which refers to interactions between different mutations. So this means that the effect of a certain mutation depends on the presence of other mutations. And in this graph, you can see that this implies, in particular, so of particular interest is the notion of sign epistasis. So this occurs if a given mutation can be beneficial, that is increasing fitness, or deleterious, that is decreasing fitness, depending on the context, that is on the starting point. And in this picture, this is indicated by the fact that the arrows on opposite edges are now no longer pointing in the same direction, right? So if you look at this arrow here, this arrow is pointing to the left, but the parallel arrow over here is pointing to the right. This is because if you're starting here, adding a mutation in the middle increases the fitness, but if you do the same thing here, it actually decreases the fitness, right? So this is the notion of sign epistasis. And what you can immediately see is that this decreases the number of accessible paths, right? So for example, if you want to go from all zeros to all ones, and your first step goes here, then you cannot go any further, because from here on, the 2 possibilities that you have will lower your fitness, right? So this is no longer an increasing path. On the other hand, so we see that sign epistasis reduces the number of these paths, at least as long as they are directed. But it may actually increase the number of evolutionary endpoints, because now you have a peak, right? So this is now a peak in the fitness landscape, because this is a state where the fitness is higher than all its neighbors. And so if you, within this model at least, where you're only allowed to follow these monotonic pathways, once you end up here, you're stuck, right? So here, this is now a landscape with multiple peaks. And so these notions of sign epistasis and having multiple peaks are all closely related mathematically. So this is sort of the framework in which one can think about a data set such as the one that I showed you in the beginning. And as I mentioned, the reason why this is considered to be important is because it tells you something about how predictable pathways are, right? So the definition here is a pathway is accessible if the quantity of interest increases monotonically along the pathway. And you have a high predictability if there is a small but non-zero fraction of such pathways, right? So if you're sitting at some peak, you want to know along which path could I arrive at that peak, then the answer to that question is simple if there's only a few possibilities along which this could have occurred. And so this was the sort of the starting point for a certain theoretical development that I will not go into very much. I'll just show you sort of one slide with some results on the next slide, but which, this is sort of a set of interesting mathematical questions. So for example, you could ask, so this property of accessibility, how does it depend on the structure of the landscape? So on the rules according to which you generate these landscapes, how does it depend on the boundary conditions of the path? And we'll see on the next slide that the most important quantity on which this depends is actually the fitness difference that you cover over the path. So the difference between the initial fitness and the final fitness. And of course, you would like to know how typical is this situation that you have a small but non-zero fraction of pathways. And so some of the answers that we have come up with is, so if you approach this, as a statistical physicist, obviously the first thing that you will do is to generate a kind of probabilistic model. And so the simplest thing to do would be to say, okay, I take this cube here and I just put random numbers on the nodes, right? So I just assume that these fitness values are random IID random variables. And then I can ask, in this setting, what can I say about the properties of these pathways? And so that leads to a problem that we have called accessibility percolation. And I thought I wanted to at least mention it here, although it's sort of not the main topic of the talk, because it's a sort of an appealing statistical physics problem. There's a kind of review article on the theory that you can find on the web. So the idea is, first of all, and this was an idea that is very old, that goes back to Stuart Kaufman, to just assign fitness values at random to the genotypes. And then the key finding here is, and then you can ask, you take 2 points on this graph, and you ask, what is the probability that there is an accessible path? That is, what is the probability that there is a path along which these fitness values are increasing monotonically? And it turns out that this depends on the fitness quantile. So, imagine that you just choose your random numbers from a uniform distribution, then this fitness quantile is just the fitness itself. And so you pick a fitness difference beta between the initial and the final point. And then one finds generically in this problem that there's a sharp transition where this probability jumps from 0 to 1 at some critical value beta star. Now, what is beta star? The first result that was established by mathematicians in Sweden, Peter Hegarty and Anders Martinsson, is that if you consider directed path, then the threshold is essentially 1, which means that you have a chance of finding such a path only if the fitness difference is as large as possible. However, if you allow for these mutational reversions, that is these backsteps that I introduced, this increases the number of paths enormously, and so it makes it easier for accessibility to occur. And it turns out that this actually leads to a non-trivial threshold beta star, which was approved by Berestitsky, Brunet, and collaborators. And so what we have done recently is to generalize this to sequences which are not binary, right? So if you think about proteins or DNA, of course, you don't just have zeros, ones. And it turns out, as you might have expected, you might, if you have more alleles, again, accessibility becomes easier, and you find that for large A, this beta star is of order log A over A. And to come back to this predictability question, near the threshold, as usual with a critical point, so below the threshold, you have essentially no path. far above the threshold, you have a lot. And so near the threshold, you'll have typically a small number of these pathways, and predictability is therefore high. Okay, so I can maybe stop briefly and ask for questions again.
Yeah, I have a question, Joachim. This is Debashish.
Yes, hi Debashish, yes.
When you look at its percolation, so there must be some correlation length that diverges at that point, like percolation correlation length. So what is the exponent if it diverges there?
Yeah, I'm not sure. I mean, this is a good question, but I don't have a sense of that. I mean, so this is really a new problem, and we have sort of focused really on this quantity. So just the probability of existence of these paths, right? And whether or not there are sort of critical exponents associated with this problem, I frankly don't know. I think this is completely open, and I'm not aware that anyone has sort of.
Other is a comment. Maybe you can correct me if I'm saying something wrong. So basically, there are some pathways which are taking it to some kind of a dead ends, and so if there is no reversibility allowed, so they will get stuck there. So this is somewhat like percolation in a brush. So if you have a percolation in a brush and there is a sort of field, so they get stuck at this long bristles and they cannot come out. So here that is being done by the difference in the fitnesses.
Yeah. I mean, of course, one has to distinguish here. It's of course true. I mean, most of the pathways that you can take, and these are typically pathways that you would find if you try to do this by a local search, right? So if you think about something like a random walker that moves along this graph and tries to go uphill in each step, those and this is something that that Kavita and others have also worked a lot on. These are these so-called adaptive walks. They tend to get stuck very quickly. But these paths that we're considering here, here we're sort of asking a slightly different question. We're asking, is there such a path? This doesn't mean that it will be possible to find it, but what these paths typically have to do, they have to be, because you basically have to cover this fitness difference beta, and so the paths that have the best chance of doing that are those that on each step only improve a little bit, right? So you don't want to go very, very steep, because then you will certainly get stuck. So you want to distribute the fitness difference as evenly as possible across the path. And so those are the paths that are contributing here.
Oh, okay. I'll ask later at the end.
Yeah. Okay. Yeah.
So Joachim, I have one question. Kavita here. Yeah. So this is for these random landscapes, right? All.
Right.
Yeah. What about the correlated ones? So it should be more easily accessible, right? For smoother.
Yeah. So right. So there's this is also in this review, these things are also discussed to some extent. Yeah. So you could put correlations. But that doesn't necessarily make things more accessible. So there's another result and this is, I should say, this is mostly work by my student Benjamin Schmiegelt, who has gotten delved very deeply into the mathematics of this problem. So for example, the NK model, which is a famous model for correlated landscapes, for the NK model, the accessibility is essentially always 0. So it's much less accessible than the random case. At least if you keep k fixed and you take the number of sites to infinity. So this is not, and this has sort of to do with the connectivity of the interaction graph, really. So it's not straightforward. I mean, of course, there are sort of, on the other hand, if you do what we call the Rough-Mount-Fuji model, that is, if you add a kind of general average slope to the landscape, then accessibility becomes much bigger. So it depends on how you introduce the correlations.
So in the NK model, if I decrease k, accessibility does not necessarily increase. Is that right?
That's right. Yes, that's right.
Yeah. Okay.
Yeah.
Thank you.
Okay. Are there questions in the chat? I didn't look.
No question in the chat
okay,
yet.
Okay. Very good. So let's go on. Very good. Okay, so now let's go back a bit to the real world. So we want to look at now this TEM-1 resistance landscape, and I showed you already one example of a piece of that resistance landscape from the experiment of Dan Weinreich. And so the first thing that we did together with a group of Arjan de Visser, so this was mainly work by Martin Schenk, a postdoc in the group, was to try to find more of these resistant mutations, right? So essentially, so it turns out this enzyme has about 2,500 possible point mutations. And so what you can do is to, just by mutagenesis, you try to create as many mutations as possible, and then you test them whether they increase resistance against cefotaxime. And in that way, Martin identified 48 different mutations and characterized them, so measured the resistance that they confer. And this panel here is a certain depiction of the histogram of the effect sizes. So this S is a kind of effective selection coefficient that one can compute from the resistance measurements. And these are the histograms. Now this looks, it's not very clear what this means. Now it turns out that in this field, a popular way of analyzing this kind of data is using extreme value theory and try to fit the data to a certain extreme value distribution. And this is sort of what is shown here. So there's something called the extreme value index, which is called kappa, that you can try to infer from the data. And what you find here is that at least at a larger concentration, so these different colors correspond to different concentrations, this kappa is about 1. And this actually means that the distribution of these values is very heavy-tailed. So it's a power law which is sort of on the edge of not being normalizable. So this is interesting because it was the first example of that kind that was found empirically. It seems to be typical for systems under very strong selection. So if you impose this antibiotic selection pressure, then there are only a small number of mutations that have a much bigger effect than all the others, and that leads then effectively to this behavior. But now, so here we're sort of mostly interested in these pathways. So what can we now do with these 48 mutations? Of course, as a theorist, you might say, okay, you go to your experimental colleague and you say, please create for me 2 to the 48 different mutants. Okay, now this, of course, your colleague cannot do because this is 10 to the 14 mutants, so this is unfeasible, and it will remain unfeasible, I think, for a long time. And so in order to make progress, you have to choose a subset of mutations. Now, one thing that we have sort of learned from studies in this area, and this is a review that we wrote a couple of years ago about the state of the field, is that which subset you choose will bias the structure of the landscape that you see. And there are sort of different choices. So in this particular experiment, all the mutations that we look at are by themselves beneficial because this is how they were chosen. Of course, you could also, and people have done that, you could also construct landscapes from mutations that are singly deleterious. What is very important here is the difference between mutations chosen for individual or collective effects. And that's, I will get back to that on the next slide. Another thing that you could do is to say, okay, I'm looking at a population evolving in time, and I combine mutations that actually occur along an adaptive trajectory. Now, in this particular work, and this is again a work by Martin Schenk and Ivan Sendro in my group, was to pick out of these 48 mutants, we picked 3 subsets of size 4. First of all, we looked at the mutations of strongest effect. All right, so this is sort of natural. You just look at the most important ones. Then we looked at 4 mutations that had a relatively weak effect, sort of more typical effects. And finally, we looked at 4 synonymous mutations, because it turns out that out of these 48 mutations here, 10 of them were synonymous. Now, synonymous means that these mutations change the DNA, but they do not change the amino acid, right? So you know that the genetic code is redundant. So each amino acid is encoded by 3 nucleotides. But because there are 64 different triples of nucleotides and only 20 amino acids. This code is redundant, so you can change the nucleotides without changing an amino acid, and this is then a synonymous mutation. And it turns out that also synonymous mutations can have effects. I will get back to that in a minute. Okay, so what I want to emphasize now here is this difference between mutations chosen for individual and collective effects. And this is illustrated on this slide. So what you now see here are depictions of 3 of these 4-dimensional landscapes, right? So on each of these pictures, you have on the x-axis the number of mutations. So this is our TEM-1 wild type. These are the single mutants, the double mutant, the triple mutant, and the single 4-fold mutant. This picture is for largest effect mutations. This is for the 4 mutations of typical effect. And this is, once again, the landscape of Daniel Weinreich, restricted now to those mutations which are not in the coding region, which is 4. Okay. And on the x-axis, on the y-axis, you have the increase in resistance. And what you immediately see is that the shapes are very different. So these 2 pictures have essentially the same shape. This one has a completely different shape. And what this shape means is that, okay, so the first step is always uphill, right? You make a mutation, and you know, you've selected these for their individual effects. If you combine 2 of them, that at least in some cases it still increases the resistance. But if you combine more than 2, then resistance goes down again. And this is the same in this picture. Whereas here, the trend is always uphill, and there's even a slight positive curvature. And the difference between these 2 cases is, of course, that here you started from these individual mutations, which were known to have a beneficial effect, whereas in the Weinreich experiment, as I told you, you start from the endpoint, right? So this is like in classical mechanics, when you calculate the trajectory of a particle, you can specify the initial position and the initial velocity, or you can specify the initial position and the final position, right? And so what you see here is that if you choose the mutations for the individual effects, they tend to interact negatively, so that if you put too many of them together, the benefit decreases again. Whereas in this case, the mutations were chosen with hindsight, and their collective effect is much stronger. And so I like to use this analogy that this is a bit like if you're hiking in the mountains, right? So you're hiking in the mountains and you want to climb a peak. If you do this on a clear day, you can see where the peak is, and you can sort of make your way, right? So I'm assuming that you don't have GPS and you don't have a map, but you can sort of see where the peak is. You know, these populations evolving in fitness landscapes don't have GPS and they don't have maps, so they have to find their way somehow. So you can find your way to the peak. On the other hand, if the day is very foggy, you don't see the peak, you don't know where it is, then the best you might think of doing is to try to go uphill as steeply as possible, right? So you look around you, you see, okay, this is a steep slope, so let's go up there. Anyone who has ever, hiked in the mountains knows that way you will not find the peak. And this is sort of what these populations are doing. So they are going uphill, as strongly as possible, but they will not find a very good peak. You can see that the resistance level of this, and this is a log scale, so the resistance level here is not reached by these other combinations of mutants. And I think this is a very generic effect, and it has actually also been seen in other experiments. So this is an experiment by Claudia Bank and collaborators in yeast, which is a larger scale experiment. So this studied several hundred combinatorial mutants combining 13 mutations in a certain protein in yeast. And this is sort of, so these mutations, the pathways here covered up to 6 mutations. This is the whole data set, and you can sort of see this is growth rate. You can see that generically, as you keep on adding more mutations, you go down. In some cases, you can go up. And this yellow subset here is now what you get from combining those mutations that eventually lead to the fittest type, which is this 4-fold mutant up here. Whereas if you combine the mutations that initially have the highest, the largest benefit, which are these green ones, they tend to go down again, right? So you have a similar picture In one case, you have positive curvature. In the other case, you have negative curvature. Finally, let me say a little bit about the synonymous resistance mutations. So this we published fairly recently. So these are now mutations that don't change the amino acid, so they don't really change the structure of the protein. They can nevertheless have an effect on resistance because they change the way in which the protein is being produced, right? So you know that proteins are being translated from the mRNA. And the efficiency of this translation process and the accuracy of the translation process depends on not only on the amino acid sequence, but also on the nucleotide sequence. And so in this picture, you see, this is sort of the landscape. So here you see these symbols. This is again our TEM-1, and these are the different mutations depicted according to the amino acid and the position. And then there's a star because the amino acid is actually not replaced by anything else. So this is at position 9, this is at position 17, 87, and 89. And you see that these are increasing resistance by up to a factor of 3. I mean, these don't have huge effects, but they do have a clear effect on the resistance. This is again a picture showing resistance as a function of the number of mutations. And this panel here shows that resistance is sort of correlated to expression level, which you can measure separately. What is striking about this, so first of all, this landscape also displays strong interaction. So you also here have many mutations that have a negative effect in certain backgrounds and a positive effect in other backgrounds. But this landscape also appears to show a kind of layered structure where certain subsets tend to have, within the measurement error, tend to have the same resistance level. And we have recently developed a kind of theory for this, which I think Debashish might be interested in, which is related to exclusion processes and the dynamics of translation. So we think that this kind of data could be explained by a model of translation with certain translational bottlenecks, that is, certain codons that are slow and that therefore lead to traffic jams. But this is, again, I just wanted to mention it. If you're interested in this, you can look at this paper. Okay, so this leads me to the last part, but before I get to that, maybe I should again ask for questions. Questions from anyone?
Mike, I'll wait for the, asking questions at the end. So I have some
Okay,
questions.
okay, please. Yeah, that's fine.
That will take a little longer, so I will wait till the end.
Okay, so right. So if there's nothing, and I don't think there's anything in the chat, so then let me get to the last part. So this is now, sorry again, I'm messing up with my clicking here. This is now recent work. This was published in the summer in eLife, and this is mainly the work of Suman Das, who joined us from Bangalore, actually, and is now a postdoc in the project in Cologne. And so this is now adding sort of a level of realism by introducing specifically the concentration of antibiotic as a parameter. And this leads me to this issue of dose response curve. So I mentioned already that the basic way in which you can quantify resistance is by measuring the growth rate as a function of antibiotic concentration, right? So this is sort of the dose response curve. And so what this picture here shows are two dose response curves, one for the initial strain, the wild type, and another one for a resistant mutant, right? And so you see, and so the MIC is essentially the point where this curve crosses 0. So the MIC of the susceptible strain, of the wild type strain, is here, and this is the MIC of the resistant strain. So this is sort of the typical picture. Now, it turns out that in many cases, these resistance mutations have a cost, as you might think, because otherwise, if they didn't, then bacteria would already have them. And so this means that if they increase resistance, they also decrease the growth rate in the absence of antibiotic. And this is a quantity that we here call the null fitness. And you can see that this is the case here. So the red line is a little bit below the blue one at 0 concentration. And at some point, these two curves therefore cross, right? And so the consequence of this is that in this concentration range, the mutant cannot grow because it has a lower growth rate than the susceptible strain, right? So typically you will have a population of susceptible bacteria, and the mutant maybe appears in a couple of them, and so here they will not have a chance. But in this regime here, the mutant is growing faster than the susceptible strain, and therefore here the mutant can actually be selected for. And therefore, this concentration, this crossing concentration, is called the minimal selective concentration. And this is actually, was a fairly recent insight, and this has been promoted in particular by Dan Anderson in Sweden. So traditionally, people believed that resistance evolution was only relevant at concentrations that are above the MIC of the susceptible strain. So only at concentrations where the susceptible strain doesn't grow at all anymore can resistance evolve. But of course, you see from this picture, which is a very simple evolutionary picture, that the resistant strain can outcompete the susceptible strain already here, even if the susceptible strain is still growing. And therefore, the mutant selection window is much broader, right? So it's this entire interval, and this has important consequences. For example, if you worry about the evolution of antibiotic resistance in the environment, right, where you can have rather low concentrations and nevertheless resistant mutations can appear. And so we were interested in how the selective conditions, and in particular these fitness landscapes that I have shown to you, how they change as a function of concentration. And it turns out, and now this might seem like a very difficult problem, because it would mean that you would not only have to measure the MIC for all the mutants, but you would have to measure each mutant over a range of concentrations. But it turns out that the problem is not as complicated as you might think, at least in some cases, because there are two simplifying observations. So the first one is that in some cases, these dose response curves display scaling. Now, the scaling, of course, for statistical physicists is something that we're very familiar with, and this is based on data that was obtained by the group in Edinburgh. This is now a different system. So these are E. coli bacteria subjected to an antibiotic called ciprofloxacin, which is an antibiotic that targets the DNA gyrase. And these are dose response curves. So here you have the growth rate, here you have the concentration of the antibiotic, and this is now a bunch of mutations. So this is, again, the black one is the wild type, and then you have a bunch of single and double mutations. And so you can see that by and large, most of them actually have this trade-off. So there's a lower growth rate at 0 concentration combined with higher fitness. There's one exception here, this purple one, which actually will also be excluded from the subsequent analysis. Now, the observation of Bartek and Rosalind was that if you now rescale these curves, as you naturally would do, so you scale the concentration, let's say by what we call the inhibitory concentration at 50%, that is the point where the growth rate has decreased by 50%, and you rescale the growth rate by its value as 0, you get a rather nice data collapse. Okay, so this is now the collapse data. You can fit this to what is called the Hill function. This is not particularly important. The main point is that you have this collapse, which means that the shapes of these dose response curves are actually essentially the same for all the mutants, right? So that sort of reduces the complexity by quite a bit. And so this means that in order to describe this dose response curve, the only thing that you need to know is the growth rate at 0 and the IC50, okay? But of course, you still have the problem that you don't know how these quantities change when you combine several mutations. Now, there's a second observation, which is that these two quantities, which we call the marginal phenotype, so this is the growth rate at 0 and the resistance, that in many cases, these combine multiplicatively. So this is shown in this table, which is probably hard to read, but here you have, these are 4 mutations. So this is a subset of a total data set of 5 mutations. The middle one is missing. This is the purple one that I showed in the beginning. So these are 16 combinations, and what is shown here. And so now you can ask, you can measure, for example, the growth rate of a combined mutant, and you can compare this with the growth rate that you would predict from the individual constituent mutations by just multiplying their effects together, right? This is what combined multiplicatively means. And in all cases, except for the purple cases, this is true within error bars. In the purple cases, the actual growth rate is lower than the predicted one, and the actual resistance is also lower, which means that there are negative interactions between these phenotypes. But for the modeling, we'll now assume that this multiplicativity holds throughout, which is true in a good approximation. Here it turns out that these negative interactions, and for some problem or for some properties, actually don't matter much. And so this pattern was actually first observed by Dan Anderson and collaborators in a different system. At this point, we don't know how general it is, but at least there are cases where it occurs. And so in those cases, one can formulate a rather simple model that includes the entire concentration dependence. So the idea is the following: we have a certain set of resistance mutations. Each of these resistance mutations is associated with a reduction in the growth rate at 0 by a factor Ri, and an increase of resistance by a factor Mi. Right. So these are 2 numbers that characterize the mutations. Then we have a mutant again, indicated by these binary variables, and then the dose-response curve of the mutant is a scaled version of this function f of x, which is the same for all the mutants. And the scaled version has 2 parameters, so it has this characteristic concentration and the characteristic growth rate, and those are just obtained by multiplying the factors associated with the mutations that are present. Okay, so this is the model. Here resistance will be quantified by the level at which the concentration at which growth rate drops to 50%, which means that this function f of 1 that this function takes the value 1/2 at 1. And so with this model, you can basically, from these 2L numbers, you can generate the entire concentration dependent landscape for this system. Okay. And so what are the properties of this model? So now you can basically do the following. You can sort of start at 0 concentration. At 0 concentration, everything is very simple because these growth rates simply combine multiplicatively, and then you increase the concentration, and then what will happen is that these dose response curves will start to cross, right? So this is an example. So here we have, with just 2 mutations, here we have the dose response curve of the wild type. We have 2 mutants, 1 and 2, the red and the green line, and then here we have the double mutant, which is blue. Okay, and so now you see, so initially, of course, only the wild type has the highest growth rate. At this point, the wild type crosses a mutant 1. So from here on, mutant 1 is fitter than the wild type. Then the wild type crosses mutant 2, and finally it crosses the double mutant. And so at high concentrations, you see that the ordering here, so here the ordering is mutant is wild type, mutant 1, mutant 2, mutant 1, 2, and here this ordering has been reversed. And this is shown in these fitness graphs here on the right-hand side. So initially, the all 0 state is the fittest, then the 1, 0, 0, 1, and finally the 1, 1. Okay, so this model sort of predicts a certain evolution of the fitness graphs as a function of concentration. And this is, this leads to a sort of interesting combinatorial problem. It turns out that not all possible rank orders can appear in this process. What is important in terms of so if you want to evolve resistance at intermediate concentrations, you'll typically be faced with a landscape like this, that in this case now has two peaks, right? So what these multiple crossings are generating for you is a kind of increasing complexity of the landscape. So the landscape becomes sort of more and more rugged. And this you can sort of quantify. So this is now a simulation using 16 mutations with randomly distributed R's and M's. And what you can then measure is the number of peaks in the landscape. This is a possible measure of ruggedness of the landscape. And you see that this number grows. And at large concentrations, of course, it goes down again, because at large concentration, only the mutant which has all the mutations will be able to survive. And there are some quantitative laws here. So there's a typical concentration at a concentration X. The typical number of mutations is logarithmic in the concentration. The number of peaks grows exponentially with L, as is usual in these kinds of landscapes. So this crossing of these dose response curves generates a rather complex landscape. And so now you might ask, okay, so suppose now I'm at some intermediate concentration, and I want to try to evolve resistance at that concentration. Then, as we discussed before, because there are these peaks, this will probably be difficult, so the population will tend to get stuck. But here, and this I think was, for us the most surprising result. This is a mathematical statement that Suman was able to prove, which is that these landscapes have a very peculiar structure. Formally, what one finds is that certain rank orders between genotypes are forbidden. So this is just a statement that you can derive. The consequence is the following statement, that any peak genotype is accessible from all its sub and supersets. So what does this mean? So this is now a 5-dimensional hypercube in the usual notation. This is at some intermediate concentration, okay? And so now you have here, for example, a peak genotype. So this is now a state that is it's 4-dimensional, I guess. This is a state that is a peak, a local fitness maximum. And so this has certain subsets and supersets. So subsets are obviously, for example, this one, right? So if you remove this one, this would give you a subset. This is another subset. And of course, the all zero state is also a subset. A superset is what you get when you add additional ones to it, right? So these are the supersets, these are the subsets. And the statement of the theorem is that at any concentration, any peak, any local peak in the landscape is always accessible. That is, fitness always increases monotonically if you go there from any of its sub or supersets. And this means in particular that if you start from the wild type, that is the all zero sequence, you can always evolve to all the peaks along a path, right? And so if you think about this in terms of the sort of actual medical problem, this means that although these landscapes are rather complicated and you might think that evolution on these landscapes would be difficult, it's nevertheless rather easy because you have all these different pathways. And of course now, as I mentioned in the discussion with Debashish in the break, the existence of these pathways doesn't necessarily mean that the pathway is actually found by the population. So in this last slide, we looked at what is called the reachability. So you can ask, so you start the population according to some actual dynamical process. You ask, what is the probability that you will reach the fittest mutant at that particular concentration? This is this red line. And what you see is that this is relatively, this probability, even for L equal to 10, this probability remains relatively high. And it's considerably higher than, for example, so this is sort of, this is what you would get if you would assume that all the peaks were equally likely to be found, but the fittest peak is always considerably easier to find, and it has a rather high probability to be found. Okay, so let me conclude. So I've explained to you this problem of evolution of antimicrobial resistance through multiple mutational steps. We've seen in this empirical example that there's a systematic dependence on how you choose the mutations that you want to combine. In the last part, we talked about this trade-off between resistance and growth, which leads to these crossing dose response curves, and therefore to these rugged landscapes. Despite the ruggedness, however, these landscapes are remarkably accessible, and it turns out that the evolution of high levels of resistance is relatively easy. What we're currently doing with Suman and collaborator Muitin Mungan in Bonn is to consider this model using time-dependent antibiotic concentration. So now you can ask, suppose I increase the concentration and let the system evolve at the same time. This is then, an example of evolution in changing environments, and you can ask questions about the trajectories that you cover when you go back and forth in concentration and so on. And finally, let me just mention the people who did this work. So in Cologne, as I said, this has been a kind of fairly long ongoing project started by a PhD student, Jasper Franke, Stefan Nowak. Currently, Benjamin is working on this project. And then there were postdocs, Ivan Chendro, Sungmin Wang, and presently Suman. We collaborate closely with this group in Wageningen, headed by Ariane de Visser. And in Edinburgh, in this last part, we collaborated with Susan Direto, Bartek Vaslav, and Rosalind Allen. Okay, so thank you for your attention. And now I'm happy to answer more questions.
Questions? So I don't see anything in the chat yet. Debashish?
Okay, if others are not asking, let me ask my question first then. My question is regarding the synonymous, codon mutations.
Uh-huh.
So there, can you go back there? This last one that you said that there are these rare codons because of which the jumps may occur. So my question here is that which one is cause and which one is effect? Is it that the rare codons for which the amino acids are also fewer, I mean, the concentration is usually lower, is that the driving force for this synonymous mutations, or is the other way around? The synonymous mutation takes place and then the amino acid concentration has to adapt?
Yeah, I'm not sure I can answer that question.
I mean, this last one that you said that, traffic jam like thing takes place
Yeah.
when there are these rare codons. So usually it would be that the corresponding amino acid concentrations are also low. That's why these are
Right, yeah.
codons. So which one is cause and which one is effect? That's something that, I'm not clear about.
Well, I mean, here, the story here is a little bit more complicated. So the main point that is sort of surprising here, qualitatively surprising, it's not so much, okay, so you have these synonymous mutations that have some effects. And there, as you say, the typical interpretation is that you have, rare codons are translated, or codons with a low amino acid concentration are sort of translated more slowly, and this sort of leads to lower expression levels. But here the main point is not so much that the synonymous mutations change the resistance, but that they can either increase or decrease resistance depending on the presence of other mutations, right? And so what we sort of show in this paper is that this cannot be explained if you assume that expression, that the relevant quantity for expression is the current of ribosomes, which is typically what people assume, right? So you have this, you have your mRNA, it's like a track, and you have the ribosomes marching along it. And so then you say the efficiency, translation efficiency is the current. And the point is simply that if you replace a slow codon by a fast one, and this is something that I think we can show rather rigorously, then this will always increase the current, and it doesn't depend on what the other codons are doing. So if you have these mutations that increase resistance by increasing expression, then they should do that irrespective of the other mutations. And so we think that this can only be explained if the relevant quantity is not the current, but the speed of the single ribosomes, right? So this is sort of, again, something that, people know from traffic research. If you as a driver want to go from Kanpur to Lucknow, right? This is the airport.
Yeah.
If you want to, then what you care about is that you get there quick. You don't care about the current on the road, right? So even if the current is rather high, but you're constantly blocked by other people, then you're not happy. So these are not the same thing. And so we think that in order to explain this picture, you have to assume that the relevant quantity is the travel time of the ribosomes.
Yeah. Okay, I'll take a look at.
But how that really, I should say I'm relatively new to this translation business, and I think to the extent that people are looking at this more carefully empirically, I think the sort of established notions of slow and fast codons and so on, I think one discovers that everything is much more complicated and depends on a lot of other things as well.
So the concentration that you looked at, concentration dependence of fitness, that is of course the antibiotic concentration. But should the amino acid concentration be also a relevant factor? Because if that is something that can be adapted, so how fit or unfit it is depends on.
Yeah. But that's sort of a question of the physiology of the cell. So I don't actually know how that is regulated. Yes, you might think that. I understand. I mean, you might think if you have another codon, then you need to produce more of another transfer RNA, but I don't know how that is regulated. I have no idea.
Okay, thank you.
So I think there's some questions in the chat.
Okay, let me have a look at it. Right. Okay. I don't understand the purpose of rescaling. What conclusions does it help to draw? Well, it sort of reduces the complexity of the problem, right? So you basically you only you don't have to characterize each mutant by its own curve. You only have one curve, and then you have two parameters that you need to determine, right? So that's why it's helpful. It probably also means something. It probably means that in some sense these mutations act mechanistically in a similar way, or that they maybe affect the same quantity. I mean, a simple scenario would be that, and this is certainly true for some of these mutations, that they basically just change the concentration of the antibiotic in the cell, right? I mean, what some of these mutations do is that they upregulate pumps that pump the antibiotic out of the cell, and so that way they effectively sort of all affect the same thing. And then what you're actually seeing is just the growth rate dependence on the internal concentration of the cell. So that would be one scenario. So, but I think this observation of scaling is rather recent, and I don't think we know very much about what it means physiologically and where it comes from. Yeah, the next question is, what do you mean by hindsight? Okay, so what I meant by hindsight here is, so maybe I should go back, I think here, right? So the point is, as I mentioned, so here, in these experiments, the starting point were the individual mutations, right? So you generate 4 mutations, you know that they are all beneficial, and then you start putting them together. And then you see that this is good for maybe 1 or 2 steps, but beyond that, you go down again. Here, the starting point was this peak, right? So the Weinreich experiment started from basically, standing on the peak and asking, where could I have come from? This, in that sense, with hindsight, right? So you already know, you're not trying to find a good place, but you already know that you have found a good place, and you want to reconstruct the pathways along which you could have gotten there. This is what I mean. And so they were chosen for their collective effect. You see, for example, here that some of these mutations in the first step have almost no effect. So you might think these are not very useful, but they become useful when you combine them with the others. All right, and I think there was somebody raising her hand.
Yeah, Shakuntala, go ahead.
Yes.
Shakuntala? I don't know if we lost her. Okay, I'm not sure. Okay, any other question? Okay, maybe she'll come back. Okay, I just have a question about this last slide about the changing environments.
Uh-huh.
So I was just thinking like, how would I model it? So what I was thinking is that, I would just think of it as a series of sudden changes in environments to start with, right? So I start with a certain fitness landscape and suddenly the environment changes. And then I will see where the population is again, suddenly it changes, etc. But I guess that important thing would also be the time scale over which these environmental changes are happening, right? So where is that information
Yeah.
coming?
I mean, so what we're doing right now, which is of course quite simple-minded, is, if you look at, for example, this, let's say this simple picture, right? So you would say that, initially you start at at a low concentration, so the population is sitting here. And then at some point, I mean, here nothing, here it's actually rather simple. I mean, then at some point, this is no longer a peak, right? So when this is no longer a peak, you have to go somewhere else. So you basically have to do one of the mutations that now becomes available. In this case, this is the only way where you can go. So you would just sit here. And then, you raise the concentration further. A second peak appears, but you still sit up here, right? Now at this point, this is no longer a peak. So now you have to, okay, now you cannot really go anywhere. You have to go here and then you go here. So here, that's not very interesting. But if you have a bigger system, then, you can ask. So basically, we're thinking about this in terms of stability. I mean, this actually, Maiti, our collaborator, he comes from this sort of disordered systems context where people look at metastable states. And so the idea is you change your concentration, you're sitting at a particular peak, you're losing that peak, and then you have to go to another peak, right? So you have to define some dynamics, for example, a greedy adaptive walk, which takes you to a different peak, right? And that way you will basically cover, with increasing concentrations, you'll be covering some trajectory. And then you can ask, what are these trajectories? And is it, for example, reversible, that if you now change the concentration back, will you retrace your step or will you go somewhere else? So those are the kind of questions. I guess with regard to timescales, this is probably a regime where, in some sense, you're assuming that the concentration changes are slow compared to the evolutionary process, which is maybe not very realistic, but that's what we're doing.
Yeah, because for example, if it's a metastable state, it will be much, much longer. And then basically, you will just remain there until the environment changes again and the fitness landscape changes again, right?
Yeah, so metastable means that it's a peak, right? So what is sort of, but when it's no longer a peak, I mean, we're not assuming that you're trying to find at every concentration the fittest state. You just want to be at a peak. But when you're not at a peak anymore, then you have to evolve.
Okay.
So you're basically moving on the network. So there's a certain subset of states which are peaks or metastable states at some concentration, and this is sort of the network on which you're moving.
Okay, I think Shakuntala has added her question in the chat.
Okay, let me see if I can find it. Dose response curve between the resistant strain and normal strain, does the steepness matter? When the growth rate drops, does it drop with a similar steepness for the two strains? Right, maybe this, I think this probably also refers to the picture that I showed. So this is a schematic, and these are actually data. This is from this paper from the Dan Anderson group. And I guess here you would probably say that the steepness is very different, right? So I think so this should probably be the dose response curve of the mutant, and this is the susceptible strain. And so you see this looks essentially exponential, and this looks much more curved. So I think in general, these could be, the shapes could be different. In our model, we're basically assuming, and I think this is sort of, seems to be by and large true here, that the shapes are all pretty much the same. And this is, of course, the scaling assumption is assuming that there's only, the shape is the same. And so the difference between the mutants and the susceptible strain is only in terms of the single parameter, right? But as I said, this doesn't, is not necessarily always true. I think it's something that is still open for research, how general this scenario is.
Okay, so any more questions? Okay, I think there were a lot of questions anyway during the talk as well. Debashish, and anything?
So maybe I can stop sharing so that I can see you again. Okay, let me see, gallery view. Okay, very good. Yes.
Yeah.
No, I have no other question. It's just some analogy that I see. If you take this dose in the sense of any enzymatic reaction, so the substrate concentration and the rate of the reaction. So there you can have Michaelis-Menten or you can have the Hill form. So you have Hill form here. So it is only numerically obtained. There's no analytical.
Yeah, I mean the fact that it's a Hill function, I think, has no deep meaning. For some reason, people like to use Hill functions to describe those response curves. I mean, this is just something that kind of, but it's purely empirical. We do this also in other studies on antibiotic resistance. But so this Hill exponent is really just a fit parameter. So I wouldn't place any stronger significance to that. I mean, Rosalind Allen has done some work where she has actually tried to derive the shapes of these dose response curves from more mechanistic, cell physiological considerations. But we haven't done that. I think there's another. Yeah. Okay. Yes. Just thank you. Yes. So in that sense, I mean, this is, you could use other forms. There's no mechanism behind it, really.
Yeah. Okay, thank you.
Okay, so I think there are no more questions. So thanks a lot, Joachim, for this very interesting talk.
Let me thank you also, and let me say, as I mentioned, I would much have preferred to come to Bangalore, and just the last couple of times, every time I go to India, it's become a small tradition that I always bring back a small statue of Ganesha. I mean, Kavita knows that I like Ganesha. So this is the one that I picked up during my last visit to Bangalore a year ago. So I'm very sad that I cannot, I'm not getting a Ganesha for my visit this time.
The next time. Yeah,
but I hope it won't be before too long, and we can meet again. Okay.
Sure. Thank you, Joachim. And
thank you very much.
Kavita.
Yeah,
okay, thank you, Kavita. It was great seeing you, and give my regards to Justin and Shama. Okay,
yeah, we'll do that. Yes, see you.
Thank you.
Bye.
Bye.